All right, let's just review a little bit about what we were doing last week. It's extremely important. There's a couple things I want to go over on the board with you anyhow. We're looking at the creation of a protein. The creation of a protein obviously resides in the nucleotide sequence in your DNA in each of or all of your cells in your body. The first part of protein creation is known as transcription, and the process of transcription ends in the creation of an mRNA molecule, which then is going to be modified. Some part of it is going to be stripped off, some of it we're going to keep. The part of it that we keep is going to be called an exon, and then part of it is going to be deleted, that's called an intron. It then leaves the nucleus, where it travels in the endoplasmic reticulum till it reaches a cell organelle, which is known as a ribosome. Once the modified mRNA molecule lays across the surface of the ribosome has two parts. It has a lower part and an upper part. And it actually lays within the area between these two segments. And the ribosome is actually going to slide across the mRNA molecule. It's going to slide across it. Notice that there is a nucleotide sequence built into the mRNA. That nucleotide sequence that you see here is in fact a conjugate copy of the nucleotide sequence which was in the DNA, with the exception that there is no tyrosine. Instead of tyrosine, there's uracil, right? Don't make that mistake, no tyrosine. Now, as it slides along, reading the nucleotide sequence, there are a new type of RNA, which is known as transfer RNA. You can see this transfer RNA has a nucleotide sequence of UUU, right? Uracil, uracil, uracil. This transfer RNA will always be connected to an amino acid known as lysine. This transfer RNA has a sequence of ACC. It will always be attached to an amino acid known as tryptophan. This one has AAG. It'll always be attached to an amino acid known as phenylalanine. And this one, which is CUA, is always attached to an amino acid called aspartame. Okay? Of which there are how many different kinds? 20. So how many different kinds of tRNA are there? 20. Right? Each one binding to a specific amino acid. Now, if you follow what I'm about to say, you will understand protein synthesis. Let's look at this one, tryptophan. ACC, right? The conjugate of A is what? Conjugate of A is U. So this one lines up. The conjugate of A is U, and the conjugate of G is C. This one fits here, this one fits here, this one would fit, here's UUU. What would be the conjugate of UUU? AAA. Look how your body is building a protein, right? It has this beautiful template, and then these transfer RNAs simply are the correct puzzle piece that can fit in there. No other one would fit in there. As it fits in, the ribosome undergoes the process of dehydration synthesis, right? And in the process of dehydration synthesis, what am I removing? Yeah. water is coming out, and what's the name of the bond that I'm forming? Peptide bond. What am I building? You got it. I'm building the protein. Mistake proof. Mistake proof. Right? So that's exactly what's going on here. Right? That's exactly what's going on. The process of reading the code is known as transcription. Translation. Right? And that's pretty cool. Now, one of the last things that I just wanted to show you you have to write this down. There are tens of thousands of different kinds of proteins that your body makes every day, right? Every day. How does it know which one to make, right? How does it know which one to make? It's simple. What process started protein formation, right? Go all the way back. Huh? Go all the way back. Back. You activate a gene. When you activate the gene, it activates a start marker and a stop marker. Right? One gene represents one protein. Right? One gene, one protein. So in effect, what is a gene? A gene is a specific sequencing of amino acids, right? A gene, look. A gene is a specific sequencing of amino acids. So my question to you is, if I copy that section, right? And in the end, I make up some mRNA. There's enough information here to make one, what? One protein, right? One protein. And then some days I won't do that one. Some days maybe I'll do the one that's above. Because remember, this repeats itself hundreds of thousands of times, right? The DNA molecule is gigantic. So all you got to do is activate one start marker, one stop marker. The RNA polymerase, which is coming down, reads that, makes the messenger RNA. That process is called transcription. The messenger RNA lays out across the ribosome. The ribosome reads the sequencing, and that process is known as translation. The end result, you make a protein. And it makes sense. It's a beautiful process, and if all things work, it's mistake-proof, right? Unless there's something wrong with the sequence, right? Unless there's something. If you have a sequencing, and part of that sequencing is making a protein, and that protein is part of, it's called gamma globulin. It makes up, if you look at a red blood cell, you don't have to write this down, but I just want you to see something. This is a molecule, heme. It's made up of two alpha units, two alpha proteins, and it's made up of two beta proteins, right? And basically what this is responsible for is the capture of oxygen in red blood cells. But in some cases, now if this is a protein, the protein to make this molecule, hemoglobin, is contained within the DNA. But what happens if there is a genetic issue with the DNA sequencing? And some of these don't work out correctly, right? This molecule will not have the correct shape. Meaning that in some individuals, this is the shape of a red blood cell, that the red blood cell will not be able to bend the way that it normally bends, or that it will not be able to capture oxygen the proper way. What disease am I describing? I put the initials up on the board. It's sickle cell anemia, right? Sickle cell anemia. This is going to be an aberration of one of the alpha units on the hemoglobin molecule. And it won't let this blood cell bend the way that it normally does. And so therefore, when it goes to enter a small capillary, sometimes it gets stuck and it gets clogged, and it's pressure on the nerves, and that can become very painful. Right? That's a genetic issue here in the DNA sequencing.
Aren't people who have that, aren't they immune to malaria?
That's what I've heard. That's what I've heard, yes. You follow what I'm saying, right? So as long as your DNA sequencing is correct, all your proteins will be made correct. But if your DNA sequencing is incorrect, then you have a genetic or hereditary issue, and you will not make one or more proteins, right? All right. Transcription, translation, DNA, mRNA, tRNA, right? All right. So that leads us to the end of this unit that I want to work on, and that is going to be proteins, because we just made proteins, and then after this one we'll do ATP production, and we'll be ready for our first exam. Some of the things that proteins do in the body. For those of you that make index cards, that's a good one to put up there. Proteins are the workhorses of the body, meaning that they get most of the housekeeping chores done that a cell requires, right? Proteins may work as, we're going to go through each of these as we go through this semester, right? I'll go through some in this unit, and then some of them I will not. But some of the things that proteins do, they work as enzymes, they work as membrane transporters, they work as signal molecules, sometimes receptors, sometimes regulating chemical reactions, and many times they work as part of our immune system, right? We will go over the ones that I want to as we proceed through the unit. All right. Proteins exhibit specificity and affinity, right? I want you to know that. Proteins exhibit specificity and affinity, meaning proteins are selective about the molecules that they bind with, right? Proteins are selective. Look at this. This is an active site. This is something that the protein is going to bind with. Things that bind with proteins are known as ligands. Something that binds with a protein is known as a ligand. In order for a protein to bind with a ligand, right, the two must be compatible. The two must be compatible with each other. Years ago, and it may have been when you were taking high school bio, if you did, and your teacher went over enzymes and proteins, they were talking about lock and key. That the ligand was the key and the protein was the lock, and the two had to fit perfectly together. It turns out that's not true at all. It does not work that way. it has to be somewhat compatible. And it's known as induced fit, right? It's known as induced fit. Meaning, if the protein and the ligand are similar in shape, right? It's similar in shape. The ligand can be induced to fit into the active site, right? The ligand can be induced to fit into the active site. The closer the fit, the higher the affinity, right? So if I have something like this, it can be an induced fit. But I have something like this, it would be not only an induced fit, but would have a higher affinity because it would be a better fit, right? So proteins exhibit specificity, right? Proteins exhibit specificity. They are selective about the molecules that they bind with. The closer the shape, the higher the affinity, right? The closer the shape, the higher the affinity. And this is exactly what I was showing you over here. Right? Proteins exhibit degrees of affinity, and that's the degree to which the protein is attracted to the ligand. The closer the fit, the higher the affinity. Right? So if you have, and you don't have to write this down, I want to show you where I'm going with this thing. If you have a protein, and that protein has an active site, and then you have ligand 1, which looks something like that, and then you have ligand 2, which looks something like that, the protein will have a higher affinity for that one than it would for that one. So more of this will combine to look something like that. More of this will go that way than that. Proteins exhibit degrees of affinity. The degree for affinity here is high. The affinity for this one is low. And that's the way it works, right? That's the way it's going to work out. All right. Not only will proteins combine with a ligand, but they can also unbind with the ligand, right? Here we can see a binding, right? But not only do we have a binding, but we can also have an unbinding going in the opposite direction, meaning a protein binding with a ligand can be reversible. Right? Can be reversible. And we'll look at the conditions in which it is and is not reversible. We're not going to do that right now. So far, what do we know about proteins? Proteins are specific. Proteins exhibit specificity. Proteins exhibit affinity. And proteins can also work in a reversible way. Not only can they bind with a ligand, but they can also unbind from a ligand as well. All right. Some proteins must be activated, right? Some proteins need to be activated. If this is an inactive protein, one of the things we can do to make that protein active is to remove a small piece of it, and that is known as proteolytic activation. Right? That's known as proteolytic activation. Take a look at this. inactive proteins can be easily recognized because of a suffix in their name, OGEN. Here is a protein. Its name is fibrinogen. It's an inactive form of a protein known as fibrin. Look at the one next to it. Pepsin is an enzyme found in your stomach, right? The inactive form of it is known as pepsinogen, right? Sometimes you'll see a prefix known as pro, but typically not. I want you to think about something. When you cut yourself, and we'll do this in 2086, one of the end results of the reactions that occur when you cut yourself is taking inactive fibrin, fibrinogen, and making it active, right? And making it active. By removing the ogen, I wind up with an active form of protein known as fibrin. And what fibrin does is it enables a process known as hemostasis to occur, which is a blood clot. You would certainly not want fibrin activated throughout your entire body, because then all your blood would clot. But it only gets activated in that one little spot, right? The other one we're looking at here is pepsin. Pepsin is a digestive enzyme, a protein that's found in your stomach. It digests proteins. You would not want this active all the time, because then it would digest your stomach. And so what happens is we proteolytically activate it when you eat. We activate the pepsinogen, it forms pepsin, and then the pepsin helps you in your digestive process. And when that's done, it's inactivated again. So by removing a peptide snippet in a process known as proteolytic activation, we can activate a protein, right? We can activate a protein. All right. We can also activate a protein by something known as a coenzyme, right? Either a cofactor or a coenzyme. Vitamins are coenzymes. Look at this. Here we have an inactive protein, right? We have an inactive protein. It doesn't have a binding site. But by adding either a cofactor or a coenzyme here, we activate the protein by adding the active site, right? So we have a lot of proteins, and one of the ones we're going to look at is processed in the mitochondria, which is the formation of ATP from carbohydrates. There are a lot of proteins involved in these reactions, which have to be activated by vitamin B12. And we'll take a look at the role of vitamin B12 in the production of energy. A lot of times, people that are running low on energy go to the doctor and get an injection of vitamin B, usually vitamin B12, which is going to help them produce energy in their cells that produce ATP. So, we can activate a protein by adding a coenzyme vitamins. All right. All right, this is an interesting one. I want you to know this top line up here. Modulators alter protein activity. Modulators alter protein activity. This is a ligand. Right? This is a ligand. It also can fit into the active site, but it has a very special name. It's called an antagonist, right? An antagonist. An antagonist binds and occupies the active site, but it doesn't do anything. It blocks. It blocks the active site so that the normal ligand can't bind with it. It's an inhibitor, right? It's an inhibitor. That is known as competitive inhibition. Okay? It's known as competitive inhibition. An agonist is a ligand that blocks the active site and renders the protein inactive. Right? There are a lot of drugs that act this way. There are some drugs which doctors give people that have a heart condition known as beta blockers. They work just like this. There are drugs out there that doctors give people with high blood pressure, and they're called alpha blocking drugs. And these drugs block the active site on proteins that would affect either heart rate or blood pressure. And so an antagonist binds to and blocks the active site. The normal ligand is known as an agonist. I'm not going to write it there, I'll write it here. A-G-O-N-I-S-T. This is an agonist. This is an antagonist. It binds to and blocks the active site. This one would bind to and activate the active site. The agonist activates, the antagonist blocks. All right. Competitive inhibition. All right. Then we have a couple of fancy words. I want you to know this word is called allosteric modulation. The word allosteric means away from the active site, right? Away from the active site. Here, look at this. No binding site, right? No binding site. But if you add the allosteric activator, I get the formation of an active site. That's known as allosteric activation. Activation of the active site away from the active site. Right? Activation of the active site, activation of the protein away from the active site. Allosteric modulation, right? This will activate it. So then on the flip side of that coin, of course, would be allosteric inhibition, right? Here, I had an active protein. I add my allosteric inhibitor, and I remove the active site. This is an inactive protein, right? This is an inactive protein. Allosteric activation, allosteric inactivation. All of these words are going to be up on your exam. All right. Let's take a look at this one. Modulators alter protein activity. Right? Modulators alter protein activity. We looked at allosteric activation, allosteric inactivation. Now we're looking at the effect of temperature. Right? Temperature is also a modulator. Here we have increasing rate. This is the rate of enzyme activity. Notice that at or about our body temperature, enzymes have their highest rate of action. Just about at our homeostatic temperature, we have the highest rate of protein activity. Notice as temperature increases, the rate of reaction of a protein declines. And that would explain why people that run extremely high temperatures, 105, 106, 107 are in danger of dying, and that is because I have protein activity which is declining, and all of the events that proteins control will stop. On the other hand, it also explains, in some instances, how it is possible for something like a fish to get frozen solid in the ice and stay confined within the ice until the spring thaw. And that's because of the fact that all of the enzyme reactions or the protein reactions are brought down to a standstill. Same thing with a hibernating bear, right? All of the reactions within the bear's body slow down to a minimal amount because the body temperature of the bear drifts down, 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 down. All right. The other thing that is going to affect the activity of proteins is pH. Notice that as pH is within the realm of where it should be, like 7.1 to 7.4, that protein activity is going to be high. But as you go either more acidic, which is going to be below, or over 7, which is going to be alkaline, that protein activity is going to fall down. Right? Protein activity is going to fall down. So, modulators of protein activity. All right. Modulators of protein activity. One are going to be competitive inhibition with an antagonist, binding and blocking the active site. We can also activate a protein away from its active site, and that is called allosteric activation. And we can activate a protein, deactivate a protein by allosteric inhibition. We can also control protein activity by varying temperature or by varying pH. It's really kind of interesting. If you have, I want to show you something. Show you how ingenious the body is. So you have the stomach, and then you have the first part of the intestines, which is the small intestine. And you got a valve there and a valve there. So you eat protein, and protein goes into your stomach. Your body goes ahead and it activates an enzyme known as pepsin, which is secreted by cells in your stomach known as chief cells, and it also releases hydrochloric acid. Right? Turns out that pepsin works at low pH, and so you digest the protein in your stomach. Right? Your stomach goes up and back and up and back, and it makes that into something which is known as chyme. At which point, this valve opens, and the stomach constricts, and it pushes everything that was in it into the first part of the small intestine, which is known as the duodenum. Then what happens up over here is it deactivates the pepsin, stops the flow of hydrochloric acid, and the stomach does not get injured during the process. But what I did do is I pumped all of this stuff with the hydrochloric acid into the first part of the small intestine. Well, it turns out the small intestine produces chemicals which are buffers, and what these buffers do is they increase the pH back to 7.1 to 7.4. And in that case, we don't get any damage to the small intestine. It's very ingenious how this all works out. And the body works it out because pH modulates protein action. The stomach is acid, your small intestine is alkaline, right? Small intestine is alkaline. And it does this throughout the entire digestive system. All right. This one's important. Here what we're doing is we're looking at a graph. What we're looking at over here is increasing protein concentration, right? As you increase protein concentration from 1, 2, 3, 4, 5, notice that there is a corresponding increase in the rate of reaction, right? So as you increase the protein concentration, you will increase the rate of reaction, right? Up over here, what we're looking at is we are increasing the ligand concentration, right? 25, 50, and so on and so forth. Here, I have one enzyme present, 2, 3, 4. As you increase the ligand concentration, the rate increases until you reach the maximum number of proteins. When you reach the maximum number of proteins, the enzymes are said to be saturated. Right? The enzymes are said to be saturated. Let me show you something. Proteins exhibit specificity. Proteins exhibit affinity. Proteins exhibit saturation. Right? Proteins exhibit saturation. This is your kidney. And I have 4 proteins, right? Sugar enters into the urine, goes to the kidney, and then it starts its way out with the urine, right? But your body does not waste things like that. So as one box of sugar shows up, that protein takes it and puts it back. That would give me a reaction rate of 1. Then 2 boxes of sugar show up, and both boxes are taken out and put back. That would give me a reaction rate of 2. Then 3 boxes would show up, and that would give me a reaction rate of 3. And then a 4th box shows up, and that would give me a reaction rate of 4. How many proteins are occupied? All of them. If all the proteins are filled, we say it is saturated. What about a 5th box of sugar? Where's it going? Where's it going? Out in the urine. Right? Going out in the urine. And that's exactly what goes on in your kidneys all the time. Although you are not passing sugar unless you are diabetic, right? The concentration of sugar in your blood is always at a level that you do not saturate your enzymes or your proteins in your kidney. And we keep it at that level with a hormone known as insulin. And so therefore, if you're having insulin difficulties, you may have more sugar in the urine than you need. You saturate all of your protein, and then whatever sugar is extra passes out in the urine. And we'll talk about stuff like that in 2086. Notice here I had increasing sugar concentration till the number of ligands fills the total number of proteins. This is known as saturation. All proteins exhibit specificity, affinity, saturation. Right? I'll see you tomorrow morning.